Welcome friends to a new session on genomics and proteomics. In this session, we will discuss cytogenetics, chromosome painting, fluorescent in situ hybridization or FISH, microdissection and flow sorting. Cytogenetics is the study of chromosomes and their role in heredity. It deals with chromosome structure and composition, the methods used to analyze chromosomes, chromosome abnormalities associated with diseases, the role that chromosomes play in sex determination, and changes in chromosomes during evolution. It includes routine analysis of G-banded chromosomes, other cytogenetic banding techniques, as well as molecular cytogenetics such as fluorescent in situ hybridization or FISH and comparative genomic hybridization CGH. Over the past decades, versatile methods based on FISH have transformed cytogenetics into a molecular science and provided cytogeneticists with powerful new tools. Conventional cytogenetic testing or routine chromosome analysis, sometimes referred to as karyotyping, is provided for a variety of clinical applications. In FISH procedures, labeled DNA or RNA probes are hybridized with their complementary target DNA sequences on chromosomes. Chromosome painting is a term used to describe direct visualization using in situ hybridization of specific chromosomes in metaphase spreads and in interphase nuclei. Chromosome painting, competitive hybridization using entire chromosome specific libraries for chromosomes as probes and human genomic DNA as the competitor was one of the first applications of FISH. Chromosome painting coupled with FISH is now used routinely to enhance the identification of chromosomal rearrangements, the assignment of breakpoints and the determination of the origin of extra chromosomal material. These technologies have enabled the development of reverse chromosome painting, in which the paint is produced from sorted aberrant chromosomes and hybridized back onto normal metaphase spreads to identify directly the composition of the aberrant chromosome. Cytogenetic analysis by chromosome painting. Chromosome painting involves the use of fluorescent tagged chromosome specific DNA sequences to visualize specific chromosomes or chromosome segments by in situ DNA hybridization and fluorescence microscopy. Chromosome painting refers to the hybridization of fluorescently labeled chromosome specific composite probes to cytological preparations and allows the visualization of individual chromosomes in metaphase or interphase stages and the identification of both numerical and structural chromosomal aberrations with high sensitivity and specificity. The simultaneous hybridization of multiple chromosome painting probes, each tagged with a specific light emitting fluorochrome, has resulted in the differential color display of human and mouse chromosomes, which is also called color karyotyping. The whole chromosome painting probes are complex DNA probes derived from a single type of chromosome that has been usually flow sorted or microdissected, amplified and labeled by degenerate oligonucleotide polymerase chain reaction to generate a paint which highlights the entire chromosome homogeneously along its side. Through this method, chromosome arm specific paints and region specific paints can also be generated. This type of probe is most useful for clarifying cytogenetically visible structural or numerical chromosome rearrangements in metaphase, but painting probes are not helpful in the analysis of interphase cells because the signal domains are so large and diffuse. Fluorescent in situ hybridization has been used to dictate the location of specific genomic targets using probes labeled with specific fluorochromes. So, the chromosome painting is also called M-FISH or multicolor FISH. The technique allowed the detection of simple and complex chromosomal rearrangements and abnormalities by the conventional cytogenetic banding techniques. Chromosome painting was developed by research teams at Lawrence Livermore National Laboratories and Yale University. Both groups had taken advantage of the availability of cloned DNA libraries that were derived from flow sorted human chromosomes. Chromosome painting has improved the efficiency of screening cells for chromosome abnormalities in testing chemicals for mutagenicity and for rearrangements associated with tumors. Painting probes detect chromosome rearrangements. Chromosome painting probes are now available for an ever-increasing number of species, most notably for the mouse and the rat, allowing the expansion of chromosome painting analysis to animal models for human diseases. Fish techniques have been developed and applied to identify the origin of the markers and other structural chromosomal aberrations. The use of chromosome painting probes in 1, 2 or 3 color fish experiments has significantly improved the diagnosis of chromosomal aberrations. Chromosome painting can be used to identify homologous chromosome segments in different species and to map probes of different complexities and chromosome rearrangements in a single experiment. The complete karyotypes of various mammals, including primates, carnivores and artiodactyls, have been analyzed by chromosome painting in recent years. Chromosome painting probes available in liquid format are directly labeled in either a red or green fluorophore, and they can be mixed together to label a number of chromosomes in a single reaction.